First of all, I would like to thank the organizing committee, especially Visciconti and Paolo Calicetti, for inviting me to this very exciting conference. And I don't want to scare you, so but before I start, last week I was in a conference in Shanghai with my very good friend Paolo De Cusi, and this was a poster from this conference, the speakers. And I want to magnify this region, which is basically me. And then I also found out that there is another person called exactly the same name as me, and it looks like this. So I am not sure whether you meant to invite me or to invite this guy. So I am going to stick with this person. It's exactly the same name. And the Chinese is slightly different. I don't know. Exactly, thank you. So I think yesterday and this morning we heard quite a lot about the application of nanoparticles in medicine and pharmaceutical sciences. And particularly in these areas in terms of drug solubilization and in targeting, theranostics and so on. And of course, these are very, very important areas. And there are even products in the market today regarding these type of delivery systems. But what I would like to also emphasize that nanoparticles have other applications. And one of the most important applications, in my opinion, is they actually been used as a tool for investigating biological processes. And they're quite valuable in terms of using them and to understand the biological principles and phenomena in order to help with drug design and so on. And that is one area that we are actually focusing in terms of using nanoparticles as a kind of a model for microorganisms, and in terms of the immunology, how the immune system sees nanoparticles and what are the responses. And if you go back, I mean, immunology in a sense is one of the oldest nano subjects in my opinion, because the immune system for millions and billions of years has been coping with nanoparticles such as microorganisms, viruses and bacteria and so on. And of course, it's very relevant to the design of all these pharmaceutical nanoparticles, because at the end of the day, when you administer any type of particle into the systemic circulation or via any type of route of entry, you are against the immune system, because all these particles, even the stealth particles, will be recognized eventually by the immune system, and there will be a number of responses. Again, we heard quite a lot about these systems, which have been in operation for many years before the new terminology, nanomedicine, came into play. And this is basically what I call the nanoscale technology. Basically, we are exploiting the size dependency, or the small size of these entities. And at the same time, we have this fantastic new type of particle coming from the nanotechnology side, because we are now exploiting the size-dependent material properties. As you're scaling the size down, you're changing the atomic structure, particularly on the surface. So these type of materials showing different physical and chemical properties, such as light emission, conductivity, and so on. So that is basically true nanotechnology, yet these, we're talking about the nanoscale technology, just exploiting the size. Most of us basically focusing on these type of nanoparticles because they make sense, they're degradable, you can deliver drugs, and we can follow their fate. And some of us, and not particularly people coming from the material science, trying to focus on these. And again, there are some applications for it in medicine, but mostly I see it from mechanistic point or understanding kind of biological processes. And also you could make mix and match between these two and making more complex composite type of nanomaterials for different type of processes. But regardless of what you have, whether you're talking about these old school of nanoparticles or these new ones or a mixture, what we have learned from some of the clinical products, particularly the micellar systems, the liposome systems, and also the emulsions that actually reach the market, that when you infuse these type of materials into patients, some people react, not everybody. And for example, with Crema4EL, it's quite very well known. And these are ethoxylated castor oil, which actually form micellar structure of around 30 to 35 nanometer, which is due for solubilization of many drugs, and particularly paclitaxel. And again, you have the liposomes, both classical liposomes and also the pegylated liposomes. And again, there are some reactions to these kind of materials. But you could control these reactions. I'll come and describe what these reactions actually are. But you can control the extent of these reactions by controlling the rate of infusion. I mean, you can slow it down quite considerably, and then you will see the reactions may disappear. And that is particularly true with the liposome systems. In the clinic these days, they actually give pre-medication to the patient, such as steroids and antihistamines, in order to suppress some of these reactions. But in spite of this kind of precautionary medication, the reaction may still appear, and that is particularly true with this kind of Crema4EL-based systems. And the reactions were so severe with one of these agents, such as the Artesin, which is an anesthetic agent, which eventually was withdrawn from the market. And again, the problem with the Crema4EL is, I mean, the statistics suggest that it's about 2 to 3% fatality during the infusion. So basically, 1 or 2 people could actually die, or 100 during the infusion of the Crema4. But no such thing has been reported with the liposomes, and the reactions are much, much milder. Another interesting system that came into the market, especially in Japan, was these perfluorocarbon emulsions, and it was in the market for a few years for oxygen delivery during surgical procedures. And within 4 to 5 years of reaching the market, the Japanese authorities removed the products from the market because of adverse reactions. And when it came to the market, there were 2 or 3 trials in the United States. And that was done in patients with head and neck cancer. And the frequency of reaction in those type of patients were quite mild, but on the normal control individuals, they saw more adverse reactions. And I'll come back to that story and try to explain it, why I think that actually happened. Now, these are the type of the reactions you see following the infusion. I mean, one of the early signs of the adverse reactions is basically the lips start to swallow, to get bigger, you get fever, you get chill effect, and you'll see the systemic arterial pressure starts to drop, and the pulmonary pressure starts to rise. And if you don't give the pre-medication to some of these patients, virtually you may have 2 or 3 minutes to live, but fortunately it can be controlled by slowing down the infusion and so on. But the molecular basis of these reactions are quite complex, and we still poorly understand these reactions. And there has been indications that the complement system, which is the first line of the body's defense, could be responsible in initiating these type of reactions. Now, what I have to emphasize in here, the complement is not the only mechanism or only pathway that could lead to these type of reactions. But what is important in here is the prime function of the complement system in the body is pattern recognition. Okay? This is the most important role or immunological role of the complement system. And if you think about it, when we're making a delivery system, whether it's based from lipid or polymers, what we are introducing into the body is patterns. And that is one of the biggest problems, one of the biggest challenges, how we can control this in spite of having this complement system against us. Can be really prevented? Or to what extent complement system is actually playing a significant role in terms of initiating these adverse reactions? In my opinion, I think one of the biggest problems we have with all these nanomaterials that we are making is this characterization. Now, these properties, such as size, shape, and surface chemistry, they all play a significant role in terms of triggering the complement system, because it's a cascade of many proteins. We're talking about 35 to 40 soluble and membrane-bound proteins. And although we can control these days in terms of the size and the morphology quite easily by using different type of techniques and make them more and more homogeneous, we still lack sophisticated techniques that could actually tell us what is going on the surface of nanoparticles. And virtually in any batch that you're actually producing, you have a very, you have many populations of particles with different surface properties. And how to assess those and how to control those to make them uniform is extremely difficult. And that is part of the problem, I think, in terms of the adverse reaction. We do not understand the material that we're actually using, unless we could develop better tools for characterization. So that is what we're trying to do partly in the lab and also in collaboration with many other people in terms of how to really assess or characterize these type of materials, and then try to see in more subtle kind of way in terms of a single particle interaction with either the innate immunity or with the cells in terms of the late responses or signaling or different type of processes. But even if you could achieve that and translate that to in vivo and see whether the whole thing appears in there, the second problem is the validation of the animal models. Again, we don't have proper models that could resemble the human case. You could use, let's say, even any large animals, such as a pig or a dog or a monkey. But again, the responses in these animals could be very, very different from what you see in a clinical case. And to make those type of validation, again, you need to really understand the molecular basis of many type of reactions from different species. And there is no certainty. At the end, you have to go to the human and actually see what's going on and whether we can understand those processes in the human and try to translate it back into design of new animal systems. So I think this is a very important cycle in terms of the structural activity and especially the characterization and validation of the animal models. So what is the complement system? Now, some of you may be familiar with the complement. This is a very simple pathway, but from immunology point, it's a very complicated pathway. It's not as simple as it looks. But technically, you could trigger a complement system through three main pathways. One is the classical pathway, the lectin pathway, and the alternative pathway. And to do that, you have a set of recognition molecules that could bind to the surface of any invading entity, which could be a particle or a microorganism. And in terms of the classical pathway, we're talking about this very interesting molecule called C1, and it has three components. And the C1q is the key molecule. It looks like a tulip kind of protein. And the head of the C1q is highly cationic. And it likes to bind to many anionic particles, especially when you have a high density of negative charge sitting on the particle surfaces. But you could also trigger the classical pathway through antibodies. For example, antibodies can sit onto the surface of nanoparticles, and if the density is correct and the posture is correct, for example, IgGs, you need two molecules in a very close proximity of around 10 nanometer that could cross-link the C1q molecule and start triggering the pathway. Again, other molecules such as C-reactive protein could sit on the particle surface and again trigger the classical pathway through C1q binding. And by doing that, you're changing the conformation of these zymogen molecules, C1r and C1s, and then in turn you're activating the C4 and the C2 molecules, and eventually you're forming a complex called C4b2A, which is a C3 convertase. Now, I'll come back to that. Let me just describe what the other two pathways are. And what I would like to emphasize is particularly the lectin pathway. Now, the lectin pathway has two recognition molecules. One is the mannose-binding lectin, and one is a set of molecules called phycolines, which they come in three different forms, L, H, and M. Now, these recognition molecules recognize the mannose group on the surface of microorganisms. So just remember the mannose groups. But what we are seeing with many of the nanomedicine, the lectin pathway is actually triggered. And that raises the question, how? Because you don't have mannose group in many of these nanostructures. And again, once you're triggering this lectin pathway, you are activating these zymogen, which are the mannose-associated serine proteases. And again, they will start cleaving the C4 and C2 and forming an identical convertase, which eventually cleave this central protein called C3. Alternative pathway is always activated. You don't have to activate it. What we call it the alternative pathway turnover. And the reason is the water molecules can actually attack the C3, forming a metastable molecule called a C3 water complex. And that exposes a central part of the C3, which is prone to nucleophilic attack by hydroxyl group or amino groups coming from the surface. And that triggers the alternative pathway through a change in the conformation of the C3 and then binding of factor B and factor D, and forming a different type of convertase which could again start cleaving the C3. There is another molecule called properdin, which is also a stabilizer of these convertase, which is also cationic. And in fact, is the most cationic protein in the entire body. And it can also bind to many anionic surfaces and directly trigger alternative pathway or increase its turnover by accommodating the C3b, which is a metastable form of the C3. Now, the problem starts when you're cleaving the C3 molecule. When you do that, you are generating this molecule called C3b, which is covalently attached to a surface, and that is causing opsonization and recognition of the nanoparticles by the macrophage complement receptor CR1 and CR3. And in turn, C3b becomes cleaved by other kind of complement proteins, such as factor I and factor H, and you're forming an inactive C3b, which again is an opsonic molecule, and eventually you're processing this molecule further and further, and you're making a C3dG, which is a small component remaining onto nanoparticle surface, and ensures a complete clearance through the CR2, which is another complement receptor. But when you're cleaving C3, you're also generating a very small molecule called C3a, and that is an anaphylatoxin and could actually give some problems. But by doing this, you also have this amplification loop that could actually amplify the deposition of the C3b onto the surface, ensuring rapid particle clearance. But once you're forming this activated C3, you're forming another convertase formation, which is a C5 convertase, causing cleavage of the final part of the complement system, which form this complex, which can insert into a membrane. And if that is, let's say, a microorganism, it causes the lysis of the microorganism and cell death. By doing that, you are cleaving the C5 and you're generating a very small molecule, which is C5a, which is a very, very potent anaphylatoxin, maybe 10 times more potent than C3a, and that is giving the trouble in terms of the anaphylactic reactions in some patients. Now, there are many bypass mechanisms that could control the circulation half-life of these anaphylatoxin. Actually, they're rapidly cleared, but in some people it goes wrong. We don't understand why. That could be something to do with the threshold of the receptor sensitivity, because these are all transmembrane kind of receptors that work with the clearance of the C3 and the C5. And maybe people have different subsets, there could be an immunogenomic factor into it, and we are looking into that. But that is giving the problem. Also, the C5b and 9 gives a problem in terms of attacking the vascular endothelial cells and modulating their function, which could actually change the blood pressure or many other kind of responses that you see. So when you're activating complement by any type of nanoparticles, you're generating this anaphylatoxin, and they act on a wide range of blood cells, including the mast cells, the neutrophils, basophils. And if you are among one of the sensitive individuals, this ligation to the receptor causes the release of all these pro-inflammatory mediators, which could actually explain some of the side effects that you see during the acute infusion reactions. For example, if histamine is released, it can cause vasodilation, and that causes the blood pressure to drop. You have the release of leukotriene, which could actually activate neutrophils, and they go to the lung, and that causes chest tightening and difficulty of breathing, for example. Another important concept in here is that the complement system and coagulation pathway are the two brothers in arms. And thrombin can also directly cleave the C3 and the C5 molecules without activating the initial part of the complement pathway and inducing this type of anaphylatoxin. And this is very important, especially in cancer. Now, in human cancers, you have a very large amount of blood clots. And that is because of the high activity of thrombin in there. And if you induce complement activation in cancer, which was shown a few years ago by John Lambris in Pennsylvania, complement activation can induce tumor growth. And the reason is the C5a, which is basically a chemoattractant molecule to suppressive T cells. These are FOXP3 cells, which actually home into the cancer and causing, suppressing the immunity, and the cancer starts to grow. At the same time, C5a is also chemoattracting to progenitor cells from the bone marrow. They home into the cancer and starting making new blood vessels. And that causing a very important issue, raising an issue in terms of the cancer therapy. I'll come back to that is it really safe to even try to, let's say, by chemotherapy, to actually treat cancer? I'll discuss that towards the end of the talk. But the problem is, when you're making all this type of nanomedicines, whether they are polymeric based or micellar based or liposome based, you could modulate this targeting, going to different cells. From the stealth system, you could avoid clearance by macrophages, but not always. I'll come back to that again. But these materials, they're all formed, remember, from polymers, from the repeated units. And the pattern recognition is causing a severe problem. Now, we heard quite a lot that pegylation could be a very interesting way of suppressing immune recognition, but it is not. You also have a subset of macrophages. When they become primed or activated, it doesn't matter how stealth your system is. They will recognize it. They will clear it, independent of any opsonization processes. And we have shown that back in 1993, published in a series of papers in JNCI and the Journal of Leukocyte Biology. And that is something to do with changes in the receptor expression and the mobility of some of the receptors on the plasma membrane of the macrophages. So stealth system, from an immunological point, is really meaningless. I mean, you do have many virulent pathogens which are quite resistant to the recognition, but eventually they're also cleared by the immune system. Now, what we do in the lab, we try to understand the processes of complement activation by nanoparticles from a drug delivery point or from the nanomedicine point. That is very important because you could induce some severe conditions. But at the same time, we are trying to use these nanoparticles to actually understand how immune system works. And that is what is our main interest. At the same time, we are trying to understand or translate the process of complement activation as a way to characterize nanoparticles in terms of their surface properties, in order to say how this heterogeneity actually works. And it could be a very interesting tool that the immune system could be used as a kind of analytical tool to determine surface heterogeneity, and that is an area that we're also working into it. Now you have this kind of morphological conditions, morphology of particle, the size and the surface characteristics, and you can change each of them systematically to see how complement is activated. But in our experience, there is not one single parameter. It's a net effect of all these issues that we are actually facing. You cannot come and say that, look, I'm changing the size, I have something really, really small, and doesn't activate complement, what is wrong? Because you are not measuring it correctly. So you have to be very careful in terms of the measurement of the complement activation. Now, I'll give you a few examples. This is the Doxil, the clinical product, and that is the plasma of a cancer patient that received Doxil. And what has been done in here, you're actually adding a radio-labeled C3, and then you are looking for the cleavage product of the C3, and you can see immediately in the contact of plasma with these Doxil, you're getting the cleavage product of all the C3 proteins. And what is very interesting is the pattern not only shows the opsonization on the surface of the liposomes, but what is very interesting is this polymerization of the C3b components, which is exactly the same pattern as immunoaggregates. So in a sense, Doxil behaving like an immunoaggregate. Yet, we have been faced by many comments saying that by pegylation you could actually suppress this opsonization of the Doxil and protein binding. But if you take this conjugate out of the Doxil formulation, you're making exactly the same formulation but with no pegylated lipid, you do not see complement activation. Totally disappears. This component can form micelles, and when you add the micelles into the plasma, you do not see complement activation. It is a combination of these macromolecules together with the lipids in the bilayer. And the reason for this activation is this anionic charge on the oxygen phosphate. And we prove that by making a lipid conjugate. These are basically pro-ethyl lipids for the purpose of cancer therapy. But what we have done, we have taken the negative charge and we put a methyl group in this case. And if you do that and measuring the terminal marker of the complement pathway, you can see the complement activation totally disappears as you're increasing the liposome dose, and regardless of the liposome size or the amount of the conjugate you're putting into the bilayer. And again, the anaphylatoxin levels remains very low and is not activated when you are changing it to this negative charge. But the mechanism is quite complex, so I'm not going to show you that. It's been published in Phathom Journal a few years ago. But the reason is that everybody in this room, you all have antibodies against the phospholipids. Now, some of these antibodies can recognize two phosphate head groups separately, simultaneously. And when you have a lipid conjugate to a polymer, it usually sits just above the plane of the plasma, of the lipid bilayer. So basically, the posture for the antibody is correct to simultaneously bind to a phosphate group higher than one on the plane of the membrane, and that causes the triggering of the complement through the C1q binding. So what is now interesting is, if you remember yesterday, they were showing the picture of Doxil, which is kind of a rugby ball shape, right? Now, if you take this formulation that's supposedly not activating complement and load it with doxorubicin, you will see complement activation, but not to a great extent. And the reason for that is the change on the curvature of the bilayer, especially towards the edge of the liposomes. Again, causing this kind of distortion and complement activation, because now the distortion is the phosphate group at the edge of the bilayer on the corner of the liposomes. When we showed that, we were also interested, we came across a veterinary product, and that is an antibiotic solubilized in polyethylene glycol. And that is coming from the leaflet of this material. And again, you can see the reactions that have been reported in cattle. I mean, initially, this antibiotic was prescribed for pigs. And pigs are quite close in terms of the immune responses to humans. And later it was withdrawn for the pigs, and now it's prescribed only for cattle. And again, you can see the same reactions. So that raises the question, can polyethylene glycol in soluble form also activate complement system? And the answer is yes. I'm not going to show you the data. And again, that was published a few years ago in Molecular Immunology, that we show that polyethylene glycols, regardless of their molecular weight, they can induce complement activation. And the most fascinating thing about this is they're activating complement through the lectin pathway, right? And that was quite difficult to grasp. We discussed the mechanism in this paper, but in order to show you how it works, I will give you another example, and coming back to the difficulty of how to actually design an inert surface from the immune system. So what we did, we used block copolymers, such as polyoxamers and polyoxamines. We have polypropylene oxide in the center and polyethylene glycols at each corner. And you can coat polystyrene model particles very easily by physical absorption. And by doing that, you can form adsorption isotherms and try to quantify not only the thickness layer, but also the quantity of the polymer sitting on the surface. And by doing that, you will see that in the first plateau, we are talking about maybe 4,000 molecules of these polymers. And at the top plateau, we're talking about 3 times more, around 12,000. And when you do rheology in terms of apparent viscosity measurement in a kind of a right volume fraction, you will see the viscosity starts to increase from uncoated particles quite high when you reach to the first plateau. And when you come to the top plateau, you can see the viscosity starts to drop. And that was giving an indication of the polymer conformation, because initially, if you have polymers very close to the surface, i.e. in a kind of a mushroom or flat configuration, when you're increasing the shear rate, you're getting chain-chain entangling, so the viscosity goes up. But when you're forming brushes, when the particles are approaching each other, they behave like springs, so the viscosity starts to drop. And you can even see in terms of the TEM, you can see you get aggregates, and as you're going through the first plateau, you're forming hexagonal arrays, and then as you go further up, you're forming a stacking. You can even show the same thing by AFM on a tapping mode as a kind of a nanorheology. You can measure the forward angle. And again, you can see that the conformation is actually preserved even in a dry state. Now, why we did that? Remember, these particles are around 220 nanometers to start with, and the reason is very simple, because we wanted to design a system that exclusively targets spleen. And the reason is the blood flow in the spleen is different from the liver. You have the artery, and in the liver, basically, when the blood comes into the liver, basically you're going into the venous system in a direct connection, but not in the spleen. It branches into different arterioles, and the entire blood flushes into the tissue. That is in the Red-Pulp and in marginal zone. That is in human and also in the rat. Then you have the venous sinuses. These are the endothelial cells, but there are gaps between the endothelial cells around 200 nanometers. So the red blood cells, which are in the whole of the tissue, can squeeze themselves through these gaps because they're highly deformable. And we reason that if anything which is rigid and cannot squeeze through these gaps, it should be filtered around here. And where the macrophages are in the red pulp are in this region, not in the outside. So that is one classical mistake a lot of people make. The macrophages in the spleen are not in the venous sinuses. Exactly the other side, right? Which is in direct contact with the entire blood flushing into the tissue. So the question in here was, if we have a particle which is rigid and its size is bigger than these gaps between the endothelial cells, and providing that the particles are not cleared by the liver macrophages, i.e. long circulatory, then they should actually filter into the spleen. So that is why we use 220 nanometer sized particles, because they're just above the size of these slits and they are not deformable. Now when you look at the blood clearance, you can see that when you are on the first plateau, particles behaving like the uncoated one, they're rapidly cleared. But when you go just above the first plateau, they become long circulatory, and it doesn't matter where you go, it's always the same. Now look at the liver clearance, it's very high in this point, and as you go higher, it becomes much, much less, and the spleen goes higher, And in time, you can see 60% of the particles actually home to the spleen. But that is based in terms of the radioactivity. And if you do the correction, that translates to around 85 to 90%. Now, the shocking thing is, when you look by electron microscopy in the spleen, so this is the sinuses in the spleen. These are the endothelial cells. So the blood comes this way, flushes all over the red pulp in this case. And the red cells, you can see, they can squeeze themselves through these gaps and go back to the venous circulation. But the particles, we were expecting to be filtered in these regions, right? But if you look carefully, these are the macrophages, and they become loaded with these nanoparticles, which were stealth to the Kupffer cells. Right? So that's one example, that not all macrophages are the same. You do have macrophages that can actually recognize stealth particles. This is exactly the same way as malaria parasite. In a sense, we have copied malaria parasite. That was the whole intention. These are the red cells with the spherocytes of the parasite, the malaria parasite. These inclusions are very rigid and around 250 nanometer. So when this red cell squeezes through these gaps, this area pinches off from the red cells, remains in the spleen, is eaten up by the macrophages, and that is how you get splenic enlargement during malaria infection. So in a sense, by copying a microorganism, we actually designed a spleen-specific system that would target these macrophages. The receptor that recognizes these is not complement receptors, right? These particles can actually activate complement system. If you have the coating in a flat configuration, the activation is exclusively through the C1q binding. That is the classical pathway. Exactly the same as an uncoated nanoparticle. But when you go and you become long circulating at this stage or at that stage, there is a shift in complement activation from the classical pathway to the lectin pathway exclusively. And that was one of the biggest mysteries. But when you look carefully, why it does that is not so surprising. Remember, the lectin pathway, the mannose binding lectin and phycolin recognizes the mannose groups on the microorganisms. Now, this is the terminal end of the copolymer structures, polyoximers or the polyoximins. Just look at the sequence of the oxygen atom from any position that you like back to the second oxygen. It's exactly the same. When you have this polymer on the surface, basically you have many branches either in a kind of brushes coming out. So you transiently, within a nanosecond, you could actually resemble the mannose structure because of these sequences. This is the region where the L-phycolin binds, and that is the region where MBL binds. So by doing that, one of these molecules, you need one single phycolin or one single mannose binding lectin to trigger the entire cascade for you. And that is the problem. If you are doing this kind of configuration or surface engineering, how are you going to solve complement protein binding? Now, all the complement binds to the surface, you're doing the opsonization. The polymer chains prevent the binding of the C3b to the receptor on the macrophage. So that is why they're long circulating. But the problem is from the side of anaphylatoxin release, which could cause other kind of side effects. But the recognition of this particle by the macrophages is exclusively through a receptor which we call dectin-1, which is one, we have not even published that yet, but that is, the receptor can recognize these type of structures without any opsonization process. Then we started to work on carbon nanotubes, although I have no interest myself in carbon nanotubes like my very good friend Professor Henning. We are both critical of carbon nanotubes. But there are many applications that people are into looking, particularly in terms of the imaging, again, not from a medical point, but this could be very important from understanding some of the physiological processes. And we have a collaborator, Hunji Dai, at the Department of Chemistry at Stanford University. He phoned me one day and he said that, look, what is this complement? I just tried to explain to him, it's just a normal immunological pathway that could cause problem before many people. And he said that, can I send you some nanotubes that we have stabilized with PEG phospholipid, which I started to laugh on the phone. And he said, why you are laughing? I said that, okay, the activation is there, I can assure you. So he sent me these materials, and that was one of the biggest mistakes in my life. I had a graduate student at that time. He was finishing his PhD thesis. It was his last month. And he said, I will do it in 2 weeks. He stayed for another year. It took us 1 year to just make the system work in terms of the detection and sorting out all the ELISA assays for that. It is very complex. This experiment that you see in here, that was published in ACS Nano a couple of years ago, and this year actually. This simple experiment that you see in here, it actually took us 3 years to do it, this single slide. Now, to do that, to actually show the mechanism, so what we did, we took plasma from an individual which is genetically deficient in mannose binding lectin. Then we had to take the C1q out of the serum of this individual by a range of depletion techniques, which you do 10 times and you get once right without changing or damaging the complement system. And then we start to look how complement is activated by measuring a marker of both classical and the lectin pathway, and then using a very wide range of combination antibodies in order to attack the active center of the serine protease. And by doing that, we actually showed these carbon nanotubes activating complement exclusively through the binding of L-phyclin. And again, to show that the activation was processing, we inactivated the mannose activating serine protease to actually to double proof it. But we did not answer whether the mannose binding lectin is also involved. So we had to do a reverse experiment this time by taking MBL deficient serum, taking the C1q out, adding an antibody against L-phyclin, and then adding purified C1s MBL complex with the mannose activating serine protease to show that also playing a significant role. The reason why we're taking the C1q out is because when you add these antibodies, if you're forming any immunoaggregates, you will activate the complement system. That is why you have to take it out. And it's a very, very difficult job. You can buy C1q deficient or depleted serum from many sources, but I can promise you it never works. They are all defective. So don't waste your time on that. Try to purify it yourself. So by doing that, we showed that the carbon nanotube, these are the PEGylated carbon nanotube, which was formed by adsorption of PEG phospholipids. These are PEG 5000 in this case. They activate the complement system. Then we started to work on multi-wall carbon nanotube. That came from another group, but this time from Turkey. And this time, they covalently attached PEG to the carbon nanotube. And we're talking about PEG 1000 from this side. We're increasing the PEG size to PEG 4000. But they controlled the reaction in such a way that the absolute mass of PEG is the same in every single system, i.e. the area that you're covering in terms of the conformation changes from more dense to less dense, more spread if you can. So that is one system. The second system is we took the same thing, you sonicate them with PEG phospholipid for double pegylation to even cover the surface more in detail. You can see pretty good coverage. You can actually do a lot of analysis by atomic force microscopy, and that is basically the bare surface of the nanotube. The outcome is it doesn't matter what you do. Complement is still active. So we talked to Hunji Dai, and he came with a new coating system, which is quite interesting, using this polymer. And this polymer is attached to 2 PEG molecules. And you can see you're getting a much better coverage of the surface of the carbon nanotubes.
5 minutes.
Okay. And what is very interesting in here is only the first part of the complement is activated, but not the terminal pathway. Again, I'm not going through these complex positions. But what is interesting is the distancing between the 2 PEG chains. And if you try to make the distancing between the 2 PEG chain in the range of 10 angstrom, then you're getting the best exclusion for protein binding. So that would be one way of actually preventing complement activation, because you are not resembling the closed structure that is required for L-phyclin docking. Just briefly, I got only a few minutes, show you the same problem with dendrimers. These are the amino dendrimers, PAMAM dendrimers. Again, on a mole basis, you can see from generation 2 to generation 5, you're increasing complement activation. But you have the same number of the molecules in here, but not the same number of the amino group, but yet they are protonated. But if you take a generation 2 and you're increasing the quantity at this point, you are presenting the same number of the amino group as this one, and you can see complement activation goes much higher, but never approaches this. And the reason is because of the change in the shape. These are flat molecules, these are more spherical. But when you try to adjust them in terms of the same surface area, you're getting exactly the same effect in terms of anaphylatoxin and also in terms of the terminal marker. Again, we have looked at the mechanism why they do that. Again, I don't have time to go through it, but I can tell you that the activation mechanism is calcium dependent. And when you do a further analysis, again, it's lectin pathway binding. And in this case, it's only mannose binding lectin and not the L-ficolin. Blood co-polymers, again, coming back, that can actually form gels, temperature sensitive one. And coming back to the Japanese story, again, we have shown that the activation again in here is through both lectin pathway and the C1q. But what we did in here, coming back to the Japanese story, is that in Japan, they have a high level of HDL in their blood, or at least they had before McDonald moved to Japan. And HDL has a regulatory role in complement pathway, which I'm not have time to discuss. But if you increase the level of HDL cholesterol in the serum by adding purified HDL to the same level recommended by the American Heart Foundation, you're actually decreasing complement. Same thing if you add LDL by inducing a hypercholesteremia, i.e. a condition of a cancer patient, right? Because they need LDL. By doing that, you can see complement is actually reduced. And the reason is, but never eliminated, the reason is these particles interact with both chylomicrons and VLDL to make them pro-complement. But if they interact with LDL, you're forming this type of structures, which we call them remnant LDL, and that suppresses activation. And the suppression of activation is because of binding of ApoB in this case, so this type of micellar structures, or ApoA in this case. ApoA prevents the polymerization of the C9 component of the last complement protein, and that is why you don't see these effects, but yet ApoB100 can totally transform these structures and make them safe in terms of complement activation. So that is one way you could actually adjust the formulation in terms of personalization by doing a lipid profiling in the patients and when to actually give the formulation to the patient. Finally, I'll just briefly touch on this process of polycations and the cancer, which I promised you at the beginning. We are doing a very systematic study in terms of how polycations can actually induce cell death by going through all the pathways in the cell, such as mitochondria, electron transport. And we recently showed that many polycations can interfere with the activity of cytochrome C oxidase, and we have shown how the electrons are actually interfered by adding the polycations, and also the formation of these halo structures, which is a kind of a programmed necrosis, if you like, which is actually happens on the addition of this type of materials. But if you are using polycations for cancer gene therapy, or even when you're using cytotoxic agents, you're inducing cancer cell death through any of these pathways. Apoptosis, necrosis, programmed necrosis, autophagy, anarchy, and so on. Most of these pathways are related, in a sense, to mitochondria. And by doing that, you are changing the lipid profile on the cell membrane in terms of the expression of both phosphatidylserine and phosphatidylethanolamine, and also cardiolipin. And these are all pro-inflammatory lipids, can induce complement activation. And yet complement activation can induce tumor growth. So that is something very important. We need to look into it. We shouldn't rush and we not try to understand. I mean, that brings a very serious question whether the cytotoxic agents are really beneficial in cancer, or we'll show you some effect and then later we see a tumor growth because of the complement process. So in this case, we are using some inhibitors. I mean, a lot of people actually saying that glycolysis is actually upregulated in cancer cells. But there is no fault in oxidative phosphorylation mitochondria. And the reason is because cancer cells become addicted to an oncogene, such as BRAC, And if you use inhibitors of the oncogenes, you cannot resolve the problem because of the resistance in some cases. So what we are actually doing now, using some of these polycations in actually damaging the mitochondria and then giving the inhibitor of the oncogenes, which actually induces a blockage in the glycolysis. So basically, I'm going to bypass this. There's no time on that. So coming back to the summary, what I would like to emphasize in here is the complement system and the way that it recognizes many particulate matter. What you need to actually, or the take-home message is, we are dealing with about 5 or 6 sensing molecules. Yet, these combinations can recognize billions of different patterns. So the question is how? And what we are doing now is trying to understand this principle on the basis of how water is forming structures on the surface and what is the residency time of the water molecules, particularly on the hydrophobic pockets. If you can understand that, then I think we'll have some opportunities of making nanoparticles in terms of being invisible, at least to the complement side of the innate immune system. So finally, I need to pass acknowledgement for many of the postdocs in my lab and many of the PhD students contributing into this work. At the University of Manchester, Christy Hunter, which is actually making a range of very small molecule complement inhibitors for us at the moment. Thomas Anderson at the Danish Technical University. Janis Zabeni at Semmelweis University helping us with some pig studies. Tunja Daim in Gazi University about carbon nanotubes. I did talk about Hunje Dai, and also I didn't have time to talk about Samir's Mitragutri regarding the particle shape and how they influence complement system at Santa Barbara, and also to many of these funding sources. So thank you very much. I will be happy to answer your questions.
So thank you very much, Professor Moghimi, for this very interesting presentation. And the section is open for discussion. The first one.
Yeah, thank you very much. I have a very basic question. We have pegylated proteins like pegylated beta-protein, acetylene,